Hello and welcome to my educational video channel. It has been my constant endeavor to improve the quality of my videos through which I want to educate my students. Thank you very much for your support and keep supporting by subscribing to my channel. Hello and welcome back to my video channel. In this video, I will give you an account of Mendel's laws and Mendelian experiments. Mendel selected only one plant for his research, that was Pisum sativum. Mendel was the only son to his father. Mendel conducted only one kind of experiment, that is hybridization. Mendel published only one paper, that too was not very ostentatious, even though it was very impactful. Mendel had one godfather, one guardian all through his life, that was Abbot Cyril Knapp. And Mendel was troubled by one fellow all through his experiments, that was Brockus pisci, an insect pollinator of Pisum plants, which was actually spoiling his experiments. Mendel conducted monohybrid cross. Monohybrid cross is a cross between two plants differing in the alternate forms of one character. One character is the height. Height is the character. The alternate forms are tall and dwarf. So Mendel selected a tall plant, crossed it with a dwarf plant. In the F1 generation, he obtained only tall plants. But what he did was he selfed the F1 offsprings. In the F2 generation, he got tall and dwarf plants in a ratio of 3:1. 3 plants were tall and 1 plant was dwarf, the ratio. Based on this ratio, Mendel proposed two laws. One is law of dominance, the other one is law of segregation. Law of dominance states that any character will have two alternate forms, out of which one will be dominant, the other one will be recessive. Dominant character will always be expressed, whereas the recessive character will be expressed only in a double recessive condition, that is, in the absence of the factor responsible for the dominant character. Similarly, Mendel conducted dihybrid cross. A dihybrid cross is a cross between two plants differing in the alternate forms of two different characters. Character 1 is color, character 2 is shape, for example. So thus he crossed round yellow plants with wrinkled green plants. Same as in the previous case, here too he obtained in the F1 generation only round yellow plants, because round and yellow characters are dominant over wrinkled and green. He repeated the same method where he selfed the F1 offsprings. In the F2 generation, what he obtained was he obtained a ratio of 9:3:3:1. which clearly states that dihybrid cross is nothing but two monohybrid crosses done together. You can see the chart that I've given, the table that I've given later, where it explains very clearly that if you compute the results, a dihybrid cross is made up of two monohybrid crosses. So this result made Mendel to propose his one more law, third law, law of independent assortment. The summary of this law is that one character, that is the character of shape, has nothing to do with the character for color. So two different characters don't interact with each other, they assort independently of each other. That is the third law. So this is the summary of Mendel's work. Mendel proposed three laws. Law of dominance, where it says dominant characters will always be expressed and they are dominant over the recessive characters. Independent assortment is the other law, where it says one character has nothing to do with the other character. Law of segregation says that when two different alternate forms of a character come together in the form of germinal factors, which we now know as genes, in a hybrid, the hybrid carries two different forms of a character in the form of two different factors contributed by two different parents. So hybrid will carry two different factors for two different forms of a trait, and these two are together in a hybrid, but they will not blend. They will segregate independently into two different gametes. So each gamete is pure for any one form of the character. That is also called purity of gametes, law of purity of gametes. Thank you very much for watching this video. We will meet again in the next episode of genetics. Thank you.